what I'm going to try to do is to describe the development within the nervous system of using DNA as a drug. And just to begin, yes, I started this work with Ionis Pharmaceuticals, and I continue to work with them. And actually, the beginning with the genetics of human neurodegenerative diseases, I know this is not a meeting focused on that, but just to remind everybody that where genes have been implicated, either as causative or contributory to the disease, in the big diseases, Alzheimer's, Parkinson's, ALS, Huntington's, those genes are widely expressed within the cells of the nervous system, not expressed uniquely within individual classes of neurons whose dysfunction gives rise to the disease. And even more so was the discovery initially by Andy Singleton at NIH that if you inherit 3 copies instead of 2 copies of a completely normal gene that encodes the alpha-synuclein, you will develop early onset Parkinson's disease. So too much of a good thing. And then that was followed by the discovery that if you inherit 3 copies instead of 2 of a different gene, the one encoding amyloid precursor protein, you will develop Alzheimer's disease, and typically early onset. So if you knew that, then a therapy that would be directly on disease mechanism in those instances would be just lower the synthesis back down from 150% back down to about 100%. And so we thought, well, why don't we just do that? Why don't we use designer DNA drugs to target the expression of the RNAs encoded by individual genes? And now, of course, I know that for maybe the front row with the faculty, the senior faculty, they might recognize this, but I know if I ask the rest of you and say, how would you do that? All of you would tell me you're going to use an Si or an shRNA to suppress gene expression. But no, we're going to use an older approach. We're going to use a single-stranded DNA oligonucleotide, typically 18 mer, 20 mer, which if you can get it inside the target cell, and so that it can diffuse into the target cell nucleus, it can pair with the RNA from any particular gene, and, or actually the pre-mRNA of a particular gene, make an RNA-DNA hybrid for which the RNA, but not the DNA, is a substrate for a nearly ubiquitously expressed, largely intranuclear enzyme RNase H. And so it's a catalytic RNA destruction mechanism that relies on the presence of a single nuclease, which is already present in the nucleus. And over the years, the medicinal chemists have modified the DNA, both the backbone and the 2 and 5' positions of the ribose sugar, and to produce molecules that will have the kinds of properties that I'm going to show in just a moment. All right, so, but one feature of them and they've been tried multiple times for indications in the periphery, but in the nervous system, by today's mechanisms of administration, if you put them into the periphery, absolutely 0 gets into the nervous system. So in the examples I'm going to show you today, we're going to directly inject it into the cerebral spinal fluid, the fluid that bathes the brain and the spinal cord, and going to inject it in what's called intrathecally in the lower portion of the spinal cord. Now, when we tried this originally, the big problem with this kind of approach is that everybody knew it just couldn't work. And these drugs are 10 to 15 times the size of a typical drug. With those phosphates, they're heavily negatively charged. And nobody thought you could deliver these broadly throughout the nervous system by a focal injection into CSF. That's what all the pharmacology and neurology textbooks said. And we tried it anyway. And it turns out that the cells of the nervous system never read the textbook. And actually, both in rodents, initially shown in rodents and then in primates, that if you dose one of these single-stranded antisense oligonucleotides, designer DNA drugs in the base of near the base of the spinal cord, you can lower gene expression to a target gene uniformly throughout the spinal cord, and actually in most of the regions of the brain that are involved in the major human neurodegenerative diseases. And when we were originally using these pumps to continuous flow, and when a negotiation with the company, Medtronic, that really owns the worldwide market on these kinds of pumps for humans, when that melted down and we thought the project was over, I got the big team together, and the three of us, one of us decided, well, why don't we just inject it in and see how long it lasts? And what we learned is that, if you take an aspirin, you get 3 or 4 hours worth of relief. But in these drugs, the best of them, you don't get 3 or 4 hours, you don't get 3 or 4 days, you don't get 3 or 4 weeks, you get 3 to 4 months worth of efficacy from single dose injections. And so that meant pumps were not needed, you just needed injection several times a year. Okay, so the disease we started with is one called Amyotrophic Lateral Sclerosis, or ALS, or for the Brits, motor neuron disease. The salient feature of that is the premature degeneration and then death of upper and lower motor neurons. The paths of those neurons were traced by the great Ramon y Cajal more than 100 years ago, with the upper motor neurons who send their axons down the spinal cord to synapse directly, or more frequently indirectly, on the lower motor neurons, whose job it is to innervate the muscles and to trigger contraction. The disease, the beginning of the molecular era of discovery in ALS was the discovery by the geneticists that about 10% of disease is clearly inherited, and inherited in a dominant manner. And the founding paper was this one in 1993, led by Bob Brown, then at Mass General Hospital, and his consortium of clinical investigators worldwide, who discovered that mutations in superoxide dismutase, a cytoplasmic superoxide dismutase, were causative of about 10 to 20% of inherited ALS, or 2% of overall ALS. And the News and Views article in Nature accompanying that immediately suggested that these point mutations in the SOD1 gene were loss of function, encoded loss of function proteins. And we tested that by making several of the mutations, 6 of them, and showed one of the mutations was completely active as a dismutase in every assay. One was completely inactive as a dismutase in every assay, and the others were in the middle. And we took the one that was inactive and asked, well, does dismutase act? And here you can see in the News and Views article, it proposed loss of function was driving disease. So we tested that by taking this inactive mutant, making a transgene, expressing it in mice with 2 endogenous alleles, and they developed fatal motor neuron disease, age-dependent. And then we removed one of the endogenous alleles with this inactive mutant, and then we removed both. And you don't need to look at which color is which, because there's just no change in the disease course in these animals, independent whether they have normal dismutase activity or absolutely none. So that led to, after a contentious period, that led to the agreement that these mutations cause some gain of toxicity from the encoded protein. Although 30 years later, we as a research community, there are 9 prominent models for what the toxicity is, and we don't agree on what it is. But what we could agree on is if you could just lower the level of the synthesis of the mutant product, that would be good. So we set out to do that, and this is the founding use of a designer DNA drug in this case in the rodent nervous system. And indeed, you can see the level of RNA encoding this mutant superoxide dismutase in mock-injected animals. Oh, look, and near the site of injection, we can lower the RNA level in a dose and time-dependent manner, as you would imagine. And actually, wow, from injection into CSF, you can lower it everywhere. And then we dosed animals to lower mutant synthesis at disease onset. And this is a hyper-aggressive model where we're making lots of this mutant protein. And so that but when you inject them, and if you inject them with saline or a non-targeting antisense oligonucleotide, they only live about 25 days after disease onset. So I said it's hyper-aggressive. But if we dose them at onset, we could double survival after onset. And I have to say, if we could do that today in ALS, in any therapy in ALS, we would see it reported in the newspapers as a breakthrough in therapy development. So we realized that we could actually dose broadly throughout the nervous system, and that we could do so with efficacy. And just this year, that approach, now initiated in 2005, was approved in the United States. And here's the outcome of the final phase 3 trial of patients that were dosed, patients expressing this mutant SOD1. The ones in green dosed, and over the 6 months, they're losing function. And then, plus versus the dashed blue line, the placebo patients. But in the second 6 months of dosing, the disease course flattened, and actually they increased in function. 25% of them improved in function after 1 year of dosing, and that, if you talk to your local neurologist, is something that has just never been heard of in ALS, to see partial disease reversal. And indeed, when the original placebo patients were converted to drug, you can see, they began to flatten as well. So, and indeed, my former postdoc, Tim Miller, who co-led the trial, he indicated in a large practice at Washington University that they've just never seen, they can't find an example of that, and yet there it is. So we argued that we can indeed affect disease course by targeting the mutant gene. Now, there are multiple other applications, and I just mentioned one, and it's maybe the reason, it's part of the reason I got invited to this, because you saw that the other speakers, they're very accomplished people, and okay, so here it was. The most frequent genetic cause of Parkinson's disease, a big disease, is an activating mutation which activates a kinase, the LRRK2 kinase. And the world's most famous at-risk individual is Sergey Brin, the founder of Google. And he's made it quite publicly known that he is a carrier for the LRRK2 mutation. So he has a high likelihood of developing Parkinson's disease. And indeed, you could target the hyperactivated LRRK2 kinase with a conventional drug, but you could also target it using one of these designer DNA drugs. And indeed, with our partners, Ionis and Biogen, they initiated a trial in 2019 to do just that, using a designer DNA drug. Now, and I began with mentioning that indeed triploidy for alpha-synuclein is sufficient to drive early onset Parkinson's disease, and indeed alpha-synuclein protein accumulations are nearly universally found in Parkinson's disease. And so, if indeed those misaccumulated protein is a part of the causation, which it seems highly likely to be, then just to lower the synthesis and would be an on-disease mechanism therapy, and that too was initiated by Ionis and Biogen in 2020. Okay, so, and now Tom just showed you the oldest slide from 1973, and I thought I was going to get it, because here there's 1977. And so this is the 46th anniversary of the discovery of a protein named Tau. Tau misaccumulates inside the affected neurons in Alzheimer's disease and actually in chronic brain injury. And there's general agreement within the neurodegenerative disease community today that the misaccumulated, misfolded Tau is the best predictor today of cognitive decline. So you all have heard about the A-beta deposits, plaques that accumulate outside the affected neurons, but there will be several of us in the audience here who have accumulated plaques who are cognitively normal, but there's almost no one who has Tau deposits who is cognitively normal. Okay, so then the strategy would then be, why don't we just try to lower the synthesis of Tau? And indeed, that too, this year, you can see it's 2023, the phase 1 trial was reported here in Nature Medicine, and to no surprise for those of us who've done this, you can get catalytic RNA destruction in a dose and time-dependent manner. You can lower the level of Tau that is spilled by neurodegeneration into cerebrospinal fluid, either total Tau or phospho-Tau. Many think that that's the bad Tau form. But the part that was the most the most provocative is Biogen-released positron emission tomography, PET scanning of two of the patients entered into the trial. Both of these were entered for the first 6 months in the placebo arm of the trial, and you can see during that time, these tau deposits that were present when they entered the trial, and after 6 months of placebo, the deposits got bigger, brighter. So this active disease where they're depositing more and more tau, and then after 1 year of dosing, with converting from placebo phase to drug phase, the tau deposits were nearly eliminated. So I know it's either, you can look at this as either 100% of the patients got this beautiful reversal, or you've only got 2 patients. Both of those are right. So and Biogen released no information about cognition, but it tests the question, at least for some patients, that these deposits of these aggregated proteins are reversible, potentially through typical protein catabolism as you lower the synthesis of the product. Okay, so now I'd like to finish with one final example, and I know it's a meeting where it's on DNA and RNA, and so an RNA binding protein named TDP-43 has been found to, typically it's an intranuclear protein. When you lower it within the mammalian nervous system, 1,500 RNAs change in abundance or splicing pattern. So kind of a lot. But in disease, in ALS, almost 100% of ALS, for some reason, TDP-43 fails to be imported correctly into nuclei, and it accumulates in these cytoplasmic aggregates. But you can see you have this nuclear clearing, and that's true in almost this 97% of ALS, half of the second most frequent dementia called frontotemporal dementia. Okay, so what goes wrong, or what are the key genes affected by the loss of TDP-43 in the nucleus? And we tested that here using an shRNA to lower TDP-43 to 25% of its original level, and we noticed that, there was an abundant RNA that was changed even more than the TDP-43 level was changed, and it encodes a protein called Stathmin-2. This is actually an abundant RNA in human motor neurons, the 20th most abundant RNA, and work that we have done that I didn't include today. It's absolutely required, at least for cultured motor neurons, for their ability to recover from injury in vitro. So why does it turn off when TDP-43 is reduced? And we've determined that there's a cluster of TDP-43 binding sites in intron 1 of the Stathmin-2 gene here. And if you deplete TDP-43, here you see at the protein level, but to 25%, the Stathmin protein just disappears. And it does so because the RNA, the pre-mRNA, gets spliced at a cryptic splice site in intron 1 and cryptically polyadenylated just after that in what should have been intron 1, making a non-functional RNA that encodes a tiny 16 amino acid fragment of Stathmin. This happens in almost 100% of ALS patients, and we know that by using a classic RNA-seq experiment, where if you look in normal individuals or laser-captured motor neurons from individuals who died from non-neurologic disease, and you do RNA-seq and look for Stathmin-2, you see exon 5, exon 4, exon 3, exon 2, just what you would expect with 3' bias in a typical RNA-seq experiment. But if you now look from sporadic ALS patients for which there is no evidence for a genetic component, what we see is you see now the downstream exons are missing, and instead what you see is the use of the cryptic splice and polyadenylation site within intron 1, which nearly silences the protein coding exons. And that was true in 100% of the sporadic ALS patients that were tested. Okay, so does the loss of this protein, this one protein, does it really contribute to the actual pathogenesis of ALS? The first step is widely believed to be the withdrawal of the motor neuron, motor axon, from its target muscle. And so, we reduced the level of Stathman expression in a fully adult, 1-year-old mouse, where initially it's almost 100% innervation. This is what denervation would look like. And if you transiently suppress the synthesis of Stathman in these adult animals, sure enough, you get denervation. And if you chronically suppress it, you get even more denervation. You get exactly the initiating steps that would be predicted for an ALS-like disease. So we argue from that this is the loss broadly in ALS and other TDP-43 proteinopathies that the loss of Stathman is an important component to the neuronal dysfunction. And now how are we going to fix that? So in the previous examples, we were trying to silence something, and now I'm trying to restore something. And the answer comes from a different example of the use of these designer DNA drugs in human disease. And it's an example of one of the most abundant genetic diseases of children, spinal muscular atrophy. The SMA locus has the SMN1 gene, which in the patients, the severe patients, they inherit homozygous inactivation of the SMN1 gene, but humans, almost alone even among the primates, have a second gene, the SMN2 gene, that could encode the completely perfect protein, this RNA binding protein, but it gets misspliced because of a 1 nucleotide change, missing out exon 7, and makes a non-functional protein. So the strategy designed by Adrian Craner at Cold Spring Harbor and Frank Bennett, the chief scientific officer of Ionis, was why don't we see if we can define an antisense oligonucleotide, DNA oligonucleotide, that has been modified so that when paired to a target RNA is not recognized by RNase-H. Okay, so it has enough modifications that, and now if you do that and you look at cells from one of these patients with homozygously inactivated for SMN1, and looking for the SMN2 RNA, you see, yep, there it is. It's almost completely misspliced, but you can find antisense oligonucleotides that can restore that, to restore the inclusion of exon 7. And indeed, that led to the breakthrough prize for Craner and Bennett for the development of a successful therapy in spinal muscular atrophy, actually the first demonstration that a human neurodegenerative disease could truly be treated. The oldest of these kids, typically they die by age 1, having never developed muscle strength, and the oldest ones are now 7 and walking. So can we use that in ALS? And the answer is, we're going to try to block the use of the cryptic splice and polyadenylation site. And what I didn't tell you is that, yes, we've done the kinds of molecular biological manipulations that you would expect. It's the cryptic splice site that drives all of the misprocessing. If you delete the cryptic splice site, the cryptic polyadenylation site is not used. I don't quite understand all of that, but it's cryptic splicing that drives that. And now the question is, can we find ASOs that, by tiling across that region, can we find ASOs that can block the cryptic splice and, even more importantly, regenerate the full-length, the correct processing to make the full-length RNA? And indeed, there's the blue ASO, dose-dependent reduction of the cryptic splice to RNA, and the same for the red one, and even more importantly, and actually much more rarely, ASOs that can also restore the level of the authentic protein-coding Stathman2 RNA. So the discovery of Stathman2 as a target for the adult motor neuron disease ALS was in a pair of papers in 2019, from my team and from Kevin Eggen's team then at Harvard, and they're published back-to-back in 2019. We achieved ASO-mediated restoration in the mammalian nervous system earlier this year, and indeed there will be a pair of, by early next year there'll be a pair of trials. One from the Eggen team, Curalis, initiated trial this year, and we hope to get with Ionis ours initiated next year. So a 4-year time frame from gene discovery to having on-target, what we hope is on-target therapy. Okay, so, and here's the scorecard for using these designer DNA drugs in the nervous system. We know we can deliver these drugs broadly, not just in rodents, but in non-human primates. We can use them for catalytic RNA degradation or, in some cases, for altered RNA splicing. They have long-lasting efficacies, at least the good one, the best of them. It's been approved for spinal muscular atrophy, and there have been 9 trials that have gone forward in 4 different neurodegenerative diseases. The blue ones have been successful trials. The green ones are in progress, and we have a couple of unexplained failures. All right, so and here's the team. The founding team for the use of these antisense oligonucleotides, designer DNA drugs in the human nervous system, Tim Miller, now independent at Washington University, local neurologist Richard Smith, who was adamant that we try this, Melissa Macalonis, who runs my animal colony, and Frank Bennett, the chief scientific officer of Ionis. And then the final story on Stathman, that the development of what we hope are designer DNA drug therapies for TDP-43 proteinopathies, which is actually a cluster of neurodegenerative diseases. A pair of postdocs and a pair of graduate students now with me, and Clotilde Augier-Turian now at MGH, my longtime collaborator. Thanks very much.